Good day, I'm Dr. T and welcome to my office. So today I'd like to talk about sickle cell disease. Sickle cell disease is a very interesting and effective case study in understanding the role of genetics and protein interactions. It is also a horrible disease which severely impacts the lives of many individuals. The world would be better off without it. Now, to understand sickle cell disease, one does need to understand how hemoglobin functions, because this is fundamentally where the sickle cell disease is taking place. Hemoglobin is a protein. Its job is to transport oxygen from the lungs to the rest of the body. Hemoglobin does this by having within it a prosthetic group, or a part of a protein that is not actually made of amino acids, but is bound into the protein permanently. And this prosthetic group is known as a heme, hence the name. Within the heme there is an iron atom, and that iron atom is able to bind with an oxygen molecule, thus facilitating the transport of oxygen. Hemoglobin is comprised from 4 subunits. 2 of the subunits are of the alpha type, and 2 of the subunits are of the beta type. When hemoglobin binds to an oxygen, it will change its shape. This allows for a synergistic effect that improves its ability to transport oxygen. When the hemoglobin does not have oxygen, it's in what we call the tense state, which is actually less likely to bind to oxygen. Once one of the subunits binds an oxygen, that subunit transitions to the relaxed state, encouraging the other subunits to do so as well, and therefore encouraging them to pick up oxygens, because the relaxed state is much more readily able to bond oxygen than the tense state. Once the hemoglobin leaves the lungs, travels through the rest of the body, eventually it will release one of its oxygens. That subunit will tend towards the tense state, encouraging the other subunits to do the same, and in doing so releasing their oxygens, thus allowing for the efficient delivery of oxygen to the rest of the body, after which they can be returned to the lungs to complete the cycle over and over again. With sickle cell disease, there is a single mutation of a single base pair within the individual's DNA. This single mutation causes an amino acid located on the outside of subunit beta to be replaced from its original hydrophilic amino acid, as one would expect from a globular protein, to a hydrophobic amino acid. And this is where the problems start. Now, it is also important to keep in mind that this particular amino acid in the relaxed state is tucked into the protein a little bit more so, and therefore is less exposed. But once the protein is in the tense state, that particular amino acid sticks out a bit more. Sickle cell attacks typically occur when the individual has a lack of oxygen for various reasons. This is because more of their hemoglobin is in the tense state, and that particular amino acid is presented to the outside much more prominently. Now, the problem originates with this particular amino acid in that you have a greasy bit, but in an aqueous environment. Hydrophobic things don't like water, and if you have 2 hydrophobic things together in an aqueous environment, they'll tend to stick to each other in order to avoid the water, or more precisely to say, the water will try and avoid them due to surface interactions. Now, if you recall, hemoglobin has 4 subunits, 2 of the alpha type, which are unaffected by sickle cell disease, and 2 of the beta type. With the mutation, those are then known as hemoglobin S for sickle cell. Because there are 2 of the hemoglobin S, there are 2 greasy spots on opposite sides of the hemoglobin molecule. This means that it can bind to another hemoglobin on each end, and therefore actually polymerize. The effects of this are twofold. For starters, because it is bound to another hemoglobin, and because it is bound in the tense state and transitioning to the relaxed state would break that binding, it is actually locked into the tense state. Therefore, that subunit and correspondingly the rest of the hemoglobin is no longer able to bind oxygen readily, which of course is the whole point of hemoglobin. The other problem is even more insidious. Because the hemoglobins can stick to each other, they can form large fibrous clumps, effectively polymerizing them. These are going to be relatively linear in nature and will start distorting the shape of the red blood cell. Instead of being nice, somewhat donut-y shaped things, although without a true hole in the middle, they then transform into these crescent shapes, hence the name sickle cell. They look like sickles. Now, these crescent shapes do not transition through small blood vessels very readily. They get stuck and can create clogs. As you can imagine, That's really bad. You need blood transferring through your capillaries, and if it can't do so, great problems will ensue. There is currently modern work to try and use genetic modification and alter that one particular base pair. Alternatively, other ways of replacing the hemoglobin within the patient with sickle cell disease in order to permanently treat the disease, although that work is still very much in the beginnings of the research phase and is unlikely to hit mainstream anytime soon, although the sooner the better. With that said, have a wonderful day, and I'll see you in later videos.